It's great to see you in the audience here, so thank you very much for coming. And this is the first time I've actually done a hybrid event. This is me at the top here, and normally I would be talking obviously about my immunology. And so this is what you know about me, but obviously what I've been asked to do, Liz and I have been asked to do, is to talk about our careers in immunology. So really, what I want to do is give you obviously some history here, some personal experiences, perhaps not going into too much detail, but to pose some questions or allow you to think about what may have happened to me in relation to what's happening to you, and it might raise, you know, just things you want to discuss. So first of all, lifelong career in immunology, how and why did it happen to me? And that's quite an important question. And this has been a bit of a journey of me to think back over my career. So there are a few points I want to get across. And one is the challenges that I've raised. And I think one of those, there are many, but one of those is when to change jobs, you know, in your career. And promotion actually is an important thing I want to bring up, which I'll explain why it's under challenges. And then opportunities, which is the fun part of the job really, which has been in taking risks. And I mean that in the laboratory and also in your life choices, taking risks. And opportunities for new collaborations, which were completely unexpected and turned out to be wonderful, you know, wonderful, life enhancing. And then obviously to talk about the role of the BSI, and I've been a member of the BSI for many years, and really have looked to the BSI as a youngster in academic support, going to conferences, the networking, and then more recently in professional development. And I'm going to say a little bit about standing up for immunology and what I've been doing for BSI, and really to I guess, share those experiences. So what does an immunology PhD prepare you for? And obviously there are all these different sectors. And this is actually a career report that the BSI did. And following the outcomes of immunology PhDs, this time period may not be exactly right, but the report is here. So if you have a look at the breakdown, then, you know, half of immunology PhDs go into academia, the higher education sector. A significant number go into biotech industry, which is what you'd expect or predict. Publishing, policy making, consulting, journalism. And this may not be what it feels like right now. You might feel that fewer people stay in academia and more go into other very important sectors to support immunology in the UK and worldwide. And I would argue that we need immunologists in every walk of life that in order to progress society and our future. So I'm not actually an immunology PhD, so I'm just going to summarise my journey. So I did my PhD training actually in vascular biology, and I was at the Vabrum Institute. It wasn't the Vabrum Institute then, but very similar to this, a BBSRC funded institute. And I did a case studentship with Siva Gaigi. So I had the opportunity to go to a large pharmaceutical company to work for 3 months during my PhD, which was valuable because I learned very early on that I didn't think I wanted to work in pharmaceutical industry, but that's fine. So I did my PhD with John Gordon, not the immunology John Gordon, the B-cell John Gordon, but another John Gordon in vascular biology. I then went to Boston, to America, on a traveling fellowship and worked with Judith Folkman on tumor angiogenesis. And that was for a year. That was a very useful experience for lots of reasons, because you're exposed to a completely different, not a different way of doing research, but I remember going to lunchtime seminars, having peanut butter and jelly sandwiches, and you were listening to Nobel Prize winners at Harvard Medical School. It was really exciting. Now, I came back to the UK, and but I moved to Manchester. I got recruited to work with Bill Ford in Manchester as a postdoctoral research assistant on a 5-year programme grant that he had. And this is where I really combined the expertise I picked up in vascular biology and training in endothelial cell biology, and said, I remember the interview with Bill and said, I want to work on these specialised blood vessels called high endothelial venules in lymph nodes, and I want to grow the endothelial cells from HEV. And he gave me the job. And I was so excited by the opportunity, but this was also an opportunity for me to learn about his expertise, which was in T-cell trafficking. And going in vivo, everything I'd done before that would be in vitro. So I was thinking about the increased skill set I would get moving to Manchester. I then got a senior fellowship, MRC senior fellowship. I was supported by Eric Bell, and I really want to emphasize the support that I've had throughout my career. I then moved to the MRC Mill Hill, which is now part of the CRIC, was recruited by Jerry Klaus, who's a B-cell immunologist to cellular immunology, and John Scahill, the institute director, was very supportive of my career. As a group leader, I carried on my research and got into a new area, which was a fortuitous finding. I then got moved to Cardiff University. Paul Morgan was very instrumental in facilitating that move, and I just want to emphasize that Arwen Gallimore there in Cardiff has been a great collaborator, and really I can't thank all my supporters for the help that I've had during my career. And I've put where next ? And we can leave that box open. But really, that's my journey, but it all is not as it seems on paper. And these are the critical life decisions, these things that happen, which are really important in your life. Well, first of all, after I'd done my PhD and been to Boston, I came back to Cambridge and I met my husband to-be, and we decided to get married. So now there were 2 of us that needed jobs. He was training to be a medic. So it was easier for him to train anywhere in the country. So I looked for postdocs, then I moved to Manchester. And we moved together to London, and we were in London. By that time, we had a young family. My husband then got recruited to Cardiff, and there was no way that I was going to split the family up. And so I then really had to put the feelers out and find a position in Cardiff, and that's where Paul Morgan and John Scale were very helpful in that. So that's what happens in your life is really important, and it should not be hidden. I don't think it's a really important part of the narrative. So then again, it's still all is not as it seems on paper, because I certainly did not envisage a lifelong career in immunology when I was starting. I just took it one step at a time. I knew what I didn't want to do, and I didn't want to work in an office in front of a computer, which is exactly what I'm doing right now. But you know, I knew what I didn't want to do. I liked doing experiments. I like cooking. I like being creative. It's all that. So the move from vascular biology to immunology gave me really my interest in research, or my research interest for life. And I have to say, I've been very fortunate to have the supporters I've had throughout my career, and having support for you throughout your career as an individual is very important, I think, very important. So the challenges, what are the challenges I faced? Well, the first challenge that I faced really going to Manchester was that Bill was killed, the person I'd gone to work for was killed in a car crash within 3 months of us arriving. And you thought, oh my God, you've got all this planned, and then all of a sudden life events change everything. So I have to say that I was very fortunate then, because Eric Bell, who some of you may know, was in Manchester, and he took over Bill's programme grant. So thanks to the MRC for allowing that to happen. And then Eric said to me, Anne, I've no idea what you're doing, but if you want to carry on, I'll support you. So that was my first break, as it were. And so I then went for a senior fellowship with Eric's support. And then it was, do I hang on in Manchester and hope a permanent position comes up, or do I leave? The writing was on the wall in Manchester, because there'd be a change at the senior level, and beware of change at senior posts. So you may be in favour by someone who's in post, then the whole situation changes, and not you as an individual, one may no longer be in favour, or the odds are not in your favour. That happens in life, it happens, it's not just in immunology or academia. So I did look at other posts. I looked at academia and pharma, and I chose academia and moved to Mill Hill. Other challenges are about being promoted. So I was brought up as a youngster whereby people would suggest that, oh, Anne, you can do this job. Why don't you take on this responsibility, or why don't you do a little bit more? And that was fine. So I spent my life waiting for someone to touch me on the shoulder and say, well, maybe, you know, I support you, let's put you through the promotion. That did not happen. I ended up having to put myself forward for promotion, and that's something to be wary of as well, and I'm happy to talk about that. Beware of bullying. So bullying of you or anyone in your team, it happens at all levels and in unexpected ways, and again, that's something we can discuss. And then a challenge is really taking the time to enjoy your research, inspire your colleagues, and have a vision. And that is a challenge. It's a challenge for all of us. We're all trying to be creative all the time. So what are the opportunities? The opportunities that lead to new avenues. Friday, what I call Friday afternoon experiments, which are risk-taking. And that is really, I'm going to have a go at this. It's a Friday afternoon. It doesn't matter whether it works or doesn't work. And I think it's really important to try those things and to try those for yourself, independent of who your line manager is. You know, just have a go. Unplanned collaborations, and some of these for me have been consequential to moving and have been unpredicted and unexpected and really very rewarding. I will say that my partner's career in cancer genetics started after we really took off when we moved to London, so there were opportunities for my family as well. And then I've also put taking the time to enjoy research, inspire colleagues, etc., under opportunities, because it is an opportunity as well as a challenge, and something to enjoy and relish, because you don't necessarily get this in other jobs. You have now, or you will have a unique skill set with part of your practical training, your academic background. Be bold and use it to take risks. So this is a little example that I saw early on in my career, not me, but around me. Once upon a time, there was a Canadian postdoc who came actually to the UK. His question was, what controls lymph flow in sheep? Okay, it was a very specific question. It was that Bayram sheep, large animals. He met a Scottish person in the department who did a unique, not a unique assay, but a very specific assay which was measuring muscle contraction in vitro using gut bath type assay, so pharmacology. The two of them started talking. They had lots of coffee breaks, lots of discussion, lots of time to talk. So they combined their skill sets, took lymph vessels from sheep, strung them up in a gut bath and found contractility and what controlled it. They got a Nature paper. Now, the fact that it's a Nature paper is neither here nor there. The point is that they spoke to each other and tried something new and made a discovery. And that's, I think, what science is about. A couple of points I want to make. There is no such thing as a silly question. No such thing as a silly question. If you have a question, even if you think I've missed something, just ask it. You know, feel free to ask it. Let your data speak and don't be drawn in by dogmas. There are a lot of dogmas in immunology, so look at your data. And there is no sell-by date on data, images, and ideas. So just because you had an idea 5 years ago and you think, oh, well, that's an old idea, it's still a valid idea. And this is just highlighting, this is an image, an electron microscope image I took during my PhD generations ago, before you were ever thought of. And this is of a post-capillary venial, and this is one of a high endothelial venial, which I took when I moved to Manchester. And I've been using these images over and over again throughout my whole life. The next few slides, I'm sorry, there's a lot of text on this. You don't have to read it, but it's really notes for me. I really want to talk about research culture now. There's no point me talking about what it was like when I started. That's absolute history and you're not interested. So research culture now. So, and obviously I'm talking about academic careers, but this could apply if one is an immunologist in policy development for the Wellcome Trust or working in the civil service. You know, it's the same thing. There's ever more competition for what you might view as some risk-averse grants. You know, that funders are backing the horses that are going to win. They're backing the winners and perhaps not backing the people who are adventurous. I now see my students and postdocs or around me perhaps they're spending their 3 years on their research project, and then finally a paper gets published which might be 3 years after they left or longer after they left, and they end up as middle author or, you know, one of many first authors. Is that the right way to approach research or to reward research? Reams of supplemental data that is never read, basically. Why is it not in the main body of the paper? How did we get here? And this is something maybe we can discuss later. I think these are very important points. I see students and postdocs basically saying no to academia because of job insecurity, which has always been the case. Grant writing forever, which has always been the case, but I think it's got worse now. There are surveys that have been and reports saying that in fact the amount of time that everyone spends writing grants is not productive at all. You almost better to give everyone one grant to start off with and get everyone started. But there's no guaranteed support from your own institution. Obviously that varies amongst institutions. We were talking about full economic costs in nuclear funds. Do we see that as scientists? I've never seen any of that. Remuneration and pensions, I think, is becoming a real issue now, and I think academic salaries are really very, very rapidly losing their competitive, or they've lost their competitive edge quite a few years ago. And of course, there are many more options now for immunologists to move into immunology-related fields which are outside of academia, which are equally important. What has happened to the time to think and discuss experiments? Your coffee, all the coffee rooms were taken away from us, I remember. Why don't we just build this into the working day? You know, make sure we have that dedicated time. Maybe you'll all do that, I don't know. So I know in Manchester, they set up, I think, Bill Ford Friday in Manchester, and this was the idea to get everyone together to discuss ideas. What can we do together? You know, what are our skill sets? What can we do together that other people can't do? There is an increasing role of team science, as you know. We have papers, you know, very few single author papers, that Gary's just got one. We're working in teams, and then it becomes difficult. One has to really think seriously about how to manage the expectations of every member of that team to make sure they get the due recognition for their input, you know, for the work that they've done. And it's not easy. Some of our work is invisible and not accounted for or rewarded. For example, the contributions that you will all have made or will make to other people's careers, either in the form of reviewing papers and coming up with maybe a critical experiment that isn't in the paper, a constructively critical experiment which actually improves the paper. I've heard that many a time, reviewing grants, impacting on other people's career progression by training and obviously passing the baton on, and reviewing. You know, these are all very important, but they're not really accounted for, I don't think, in the way. And, you know, we can discuss how that could happen. Academic scientists are expected, I think, to work in a very commercial type sector with getting funding, but don't necessarily get the rewards. That's just a reiteration. And as I said, again, one reward is promotion, and that you may have to do for yourself. Don't wait to be promoted. Women and ethnic minorities are overcooked. I'm sure you've heard that expression before. And that's what I hear from HR departments everywhere. So rethinking research culture, what's been the impact of COVID and what might the future look like? I'm not saying I have any answers to this, but I just want to make these points. Virtual meetings and hybrid meetings like this, I think, are absolutely fantastic. You know, really, that people who can't get to have caring responsibilities can view, attend on the day or view afterwards, and then make contact with the people who presented, and can be very, very good for for networking. I think the chat box, although I think it's not working, you know, not open today, but chat boxes in general can be very leveling, because you can just pose your question, you know, no matter who you are. Whereas in person, you might feel as you're in junior stages of your career, you might feel a little reticent and you're not sure of your question, how it's going to go down, but I think the chat box dispels all of those potential barriers. Lab meetings are not easy virtual, and I personally find it difficult to inspire and lead remotely in a on screen. I personally do. I noticed during COVID that I was more stressed. I think we were all stressed, I know, but I noticed it myself, and things like that need to be resolved immediately, and that's difficult to do virtually, but it's at least it's important to be aware of it. Is there a future for hybrid meetings? Well, yes, we've got one today, so definitely I know there is a future. They are expensive though, the BSI will tell you that. And there are new opportunities for collaborations at virtual meetings, which I've come across, which I actually doubt would have happened in person. I know that sounds strange, but when you're at a conference of 1,000 people, someone's just presented that you want to meet and perhaps discuss collaboration, you might never find them in the conference after that. Whereas if you're actually in a breakout room together, you can do that. And in COVID, what I saw in response to COVID was quite a few people were really quick out the blocks. I was not. If your lab was set up to do SARS-CoV-2 work, then you could carry on research. Otherwise, I don't know about you, we were just closed down for 3 months. In Cardiff, I think did something really good for the ECR, students and postdocs, and set up a COVID journal club for all of the ECRs in Cardiff to cover, obviously, this burgeoning number of publications. And everyone got at least one publication out of it in the Oxford Open Immunology, which was a new journal that Danny Altman was leading on in Living Review. So that was, being in a react and response mode. So for the future, there are many leadership courses now available. Liz and I were comparing earlier. There was nothing for us. I'm not sort of bemoaning it, but we learned on the job. And now I think there are lots of courses available. I think you have to select them very well and select them for the right stage of your career. Otherwise, you can just spend your time going to courses and get nothing out of it. I think for the future, and learn how to negotiate your own career, like in business, with respect to salary, working conditions. We are individuals. We should not be in a tick box. Tick this box, you've done your job. I think that that actually stunts people being creative. We do need to be aware of tick box exercise in the future, because this is coming along. The Wellcome Trust has started to introduce this to how to improve research culture. And will people just learn what the funders are looking for and tick those boxes? And I think we do have to be very, very careful of that. And change, improvements in research culture absolutely need to come from the top. And that is the top at lots of levels. So you're the PI, the head of division, the school, the vice chancellor, the institute director. That's really, really important. And then, I just want to finish how to diversify your skill set. You've got all these skills of your laboratory-based work, your academic background, but what else? So are there other things that interest you? And you will have different interests outside of the work that you're doing. You might be, I don't know, netball captain or football, goal, whatever, organising a team. So in a university and any sort of higher education establishment, there are teaching and admin roles that need to be done by researchers and teachers. They can't be done by people who don't have that knowledge. And there are many opportunities for learning or applying your own skills picked up inside or outside your organisation. And obviously now there's certification for higher education teaching. So have a look at, and again, this needs to be aligned with your stage of career. And I think it's fair to say there's still much to learn. Even if you think you've reached the pinnacle of your career, there's always something to learn. People are like, to me, I have to say, sorry, I don't mean this disrespectfully, people are like experimental animals. You need to expect the unexpected and be able to respond, don't wait to be asked to do something. Nominate yourself when the opportunity arises. to do or write something that you are passionate about, even if it's a complaint. It will help you and will help your peers and colleagues. And consider the BSI Immunology news as a potential outlet for opinion pieces or experiences. That is your organisation's newsletter. And I put here, stand up for immunology, be a voice for your community and get involved with BSI outreach events. Again, I'm sure there are lots of opportunities. We know a lot about vaccine engagement. Think about standing for forum or board positions when they become available. You'll learn a lot. It takes you away from your day job, which is a good thing. And you'll meet highly motivated, inspirational people. And in fact, I got into this quite early on in my career, and I've written an article about it if you want to have a read, and I'm quite happy to talk to people about that. I think another thing that's very important is learn how to review papers and grants, and learn how to embrace new ideas, not be jealous that someone else has had a bright idea, and be constructively critical and not unnecessarily nitpicky. This will really help you in your writing and your communications. But be selective and learn to say no. You know, don't if you've been asked to do something and you think, oh, I'm flattered, I've now been recognised as an independent scientist, someone wants my opinion. If it's not your field, don't do it. Don't just do it to to tick the box. And challenge editorial decisions. I'm finding that that's important. If you've been asked to be an expert review and your opinion is being overlooked by the editor, challenge the editor. You know, that's really, really important. There are a lot of sessions, more sessions at BSI meetings and all actually congresses on careers, publishing, and many institutions, including the BSI, offer mentoring, and Gary's really sort of covered that. Find a suitable mentor for you and mentor throughout a different mentor for different stages of your career. If you don't like your mentor, find another one. Seek a second opinion like your GP. And in fact, just combining these two together, so about doing something when you're passionate about. Anne Cook and I were very, very passionate about an article that came up in was published in Nature Comms about gender of mentors and mentees, and basically didn't come out in support of female mentors. So Anne and I were so motivated to respond to this that I actually brought it up at BSI Forum, got feedback, and then we wrote this article, which is in Immunology News if you want to have a look. And that was actually a very, in the end, enjoyable piece to have been involved in and to work with a very, very good colleague. So I'm just going to finish. What does BSI Forum do? So it is the think tank which develops ideas and proposals, which gives the BSI a strong profile and voice for the whole immunology community, and that is important. The membership is representative of the society's membership, including individuals from all career stages and all specialities and all the devolved nations of the UK. That's very important. The voice of all forum members is equally valued in discussions, as is the membership. If you, as a BSI member, have something that's niggling you and you think, well, I don't know who my rep is on forum, contact the BSI office. You know, that's what they want to hear from the members. The society is its members. Otherwise, it's just the people who are, meet at board. Yeah. And the chair of forum reports to the board of trustees on forum's work. And so I have been fortunate enough to be the chair for the last 4 years. I'm stepping down this year. It's just been I have to say, it's a committee that gets things done. And that's a very rare thing to say. So just to reiterate what forum does, so notable achievements. I'm not saying they're achievements during my tenure, but ECR representation on the board of trustees. So there are 2 ECR reps on that. It was one ECR is to co-chair every parallel session of Congress. So this obviously inclusivity is very important. Carers grants for meetings was something that came out of forum, and career enhancing grants were very much pushed by Forum to the board level, and then they got approved. ECR training days for postdocs, and then piloting vaccine ambassador scheme for parents and families. And I know people on Forum have been actively involved in that and have really enjoyed it. So I think that's all I have to say. So I've probably gone on too long, but there's everything I wanted to bring up, not necessarily in perhaps a cohesive way, but I hope it will lead to some discussions later on. Thank you for your attention. Oh, I don't need oh, sorry. Oh, yes. Yeah, that's my notes.
Just switch off this one. Take it on. Yeah, or put it there. Yeah, yeah. So if people can hear me online, I'm standing close enough to Liz, I hope, with my mask on. Just realised. So yeah, just to introduce Liz to you all. So Liz and I have worked together, we're friends, and we actually really bonded for the first time at the BSI, one of the BSI meetings in Kensington Town Hall. So this is going back when the spring and autumn meetings were always in Kensington. And we had this common interest in veterinary immunology, and that's how we kind of got to know each other. So, and Liz has worked in Roslin when it was ABRO before, as Anne was saying about the Abraham Institutes kind of changing their names and so on. And then obviously within University of Edinburgh and Roslin Institute, and became part of that. So I'm really interested to hear about your experiences, Liz.
Thank you. So thank you very much for asking me to come and speak.
But you might have to do it on the screen.
Yeah, so
yeah,
just,
on the screen.
yes, no, Liz. I'll show it to you. See where it says slide 19 of 34.
Oh right, yeah, I see. So
You just click on that.
that's not me.
No, that one. If you click on the arrow,
All right, I see. Okay, so
that takes you.
okay, sorry about that. Okay, everyone. So good afternoon, everyone. As Gary said, I'm Liz Glass. I'm now retired, but I was a professor at the Roslin Institute for a number of years. And what I'm going to tell you today is about my own particular career path, which was, shall we say, the road less travelled in a long and winding path. And I hope that what it will do is spark some particular points for the discussion later with the panel. So thank you very much, Gary and Jayne, for inviting me, and for the BSI for funding this and giving me the opportunity to speak. Okay, so I'm talking about challenges and opportunities and what I experienced along the way. Now, I'm not saying necessarily that it's entirely the past is another country, but it is to some extent. But I think still many of the issues that I faced are still being faced today. So what inspired me to go into science in the first place? My primary school teacher actually said to me, you can go to university. Now, I had no idea what university was. I thought, I'm going to remember that. At secondary school, I have to say I almost chose to do Latin rather than to do sciences, And it was actually reading a book and just generally being interested in science, because I didn't come from a scientific background. I actually had an epiphany when I read this particular book. Not that it's a particularly great book, I don't suppose, but it seemed like it at the time, and it very much was a looking to the future, and told me that biology, and especially genetics, he foresaw test tube babies and all sorts of things in that book, and it blew my mind, really, basically. So although it wasn't really that unusual to be the first person to go to university at the time, it did feel like it to me. I had no role models, certainly no mentors suggesting that I went or what I did. And I decided to do biochemistry, and I went to Edinburgh University. Now, the year I started was the first year that the Students' Union Bar actually allowed female students into it. Can you imagine? I mean, really? And circled is me, if you can manage to see it. I was one of 4 women students out of 15, something like that. So that was interesting. Now, I have to say that in my undergraduate years, there was a lot of women. There were a lot of women in biology. So what happened after I got my degree, I decided I really wanted to earn a living. And I always had this idea in my head that it was important to be economically independent. So I don't know where I got that from, cosmopolitan probably, but that's the way I looked at things. And so I didn't want to do a PhD at that time. I wasn't sure what I wanted to do. So I went into industry. But I was bored, basically. Bored. There was no innovation possible, although I'd been told that there would be opportunities for research. And so I applied for a research associate post with Professor Barry Kay, and he was an allergologist. So he was working on allergies, particularly in relation to asthma and other allergies. And my role basically was to investigate the interaction between myeloid cells or macrophages and cancer. And that was a big topic at the time. And I essentially felt that I was in at the deep end, because I didn't know any immunology. We'd been taught about the structure of antibodies. And I'm going to pick up on lots of things that Anne said. There was no training, there was no, oh, this is how you do things. And in fact, my supervisor, Barry Kay, actually literally said to me, there's the lab, there are 2 PhD students in there and an honours student. On you go. And here's the project that you're going to do. So it was really great that we had an energetic and enthusiastic community of immunologists in Edinburgh. There was no course, there was no immunology honours course or anything at that time, but there was what was called the Mechnikov Club, and it was, it's now been affiliated to the BSI, but at the time was very much a local forum for lunchtime seminars, for evening seminars. And at first, I have to say, I understood nothing. But gradually over the years, I began to really get my head around it. And I have to say that when I started, I always call it the black box era, because we used to work on supernatants generated by certain methods. It wasn't like immunology today, and it was pre the real molecular biology revolution. So, and as Gary said, the BSI allowed me to meet other immunologists and led to long-term collaborations over many, many years. And I have to say, for the BSI, it's a great society to belong to, because it's so generous with giving out, for example, travel grants, just so that you can go to meetings. I had some myself, but mostly in the end, towards the end, I would be applying for or my students would be applying for money and I wasn't taking any money from them. So moving on. So I then decided halfway through this research associate job that really, actually, I could just as easily do a PhD. So I asked Barry if I could. And in fact, at one point, I'd actually thought that I would go away and do it somewhere else. But he said, no, you can do it with me and you can do it part-time. So that was great. And I got my first paper, which probably doesn't make a lot of sense to any of you, but we were looking at complement receptors on macrophages in the presence of chemotactic agents, which increased their number. And I went on to do various other papers as well. But Barry then announced he was going to London. Now, he did say to me I could stay in the pathology department and finish my PhD on my own, but I just thought that was a terrible idea. So instead, I thought, right, well, I'm going to go and find another research associate post and see where I get to. And actually, I was really lucky because I applied for this job and I got it. And Donald Weir also said to me, you can carry on if you want to. You just change the focus a bit. So I worked on other receptors, which were called lectin-like receptors at the time. And I guess that what we would now call them are pattern recognition receptors. And they were visualized using formalin-killed Staphylococcus aureus, Staphylococcus albus, in the absence of serum. So they weren't coated in any antibodies or complement. And they were inhibitable by sugars such as glucose and also anti-MHC mAb. Now, we didn't know what the structure of the MHC was at the time. And I don't really know to this day where that research would have gone, but that's what we did at the time. And then things changed. So, my PhD was very unlike PhDs today, I would say. We created our own reagents, or I did. There wasn't much interaction really with my supervisors, and there was an academics only coffee room, which you just can't imagine today. I got my PhD. My examiner was Ralph van Firth, who was the father of the reticuloendothelial system, really. And by that time, I'd published 3 papers, which actually I don't think even today is that bad. And then I won a fellowship funded by Eli Lilly and the British Diabetic Association to look at the function of these receptors in diabetes, with the idea that they might be involved in infections that diabetics are more likely to get. So I set up 2 models, I got some papers, and then what? Well, so I have to, is that what I'm going to say next? So just briefly saying, I applied for several what were called new blood lectureships. And I also applied for a full grant, building on my research to various places, but I didn't get any of them. And the British Diabetic Association did, though, give me another year. Now, I've been active in the Association of University Teachers and with 2 subgroups on inequality issues and short-term contract issues. And I felt quite strongly about this. And I never felt myself that I was discriminated against, but I think the statistics tells you everything. And I went to this conference in Edinburgh and it was reported on in the Scotsman, and actually I got my name in the Scotsman, so and I kept the cutting. There was mention that these new blood posts had done nothing to bring more women into the universities. So I'm not quite sure how many, what the numbers were at the time. But we as a committee on inequality issues had shown that there were very few women, full stop, above the lecturer level, and it just went down and down and down. And I mean, it's still to some extent like that today. And there were very few women being promoted, very few. So that's the kind of background. So then I applied for a job as an immunogeneticist at the Animal Breeding Research Organisation, which later morphed into the Roslin Institute. I was appointed in July 1984, although I didn't start straight away because I wanted to finish my fellowship off first. And in the meantime, I had a phone call from my new boss, Roger Spooner, saying that my job had gone live, and this meant that the post could be filled by someone else being made redundant from another institute. And this continued to happen to me throughout my career. Well, not throughout my career, but it did happen to me during my career again and again, that we would have these additional uncertainties. So the Animal Breeding Research Organisation was focused on animal breeding and genetics. And I think I was being rather naive and didn't ask enough questions actually when I took this post, because it was tough and very challenging. Immunogenetics as a group didn't really fit into this institute, and I didn't appreciate that at the time. There was a lot of ongoing economic uncertainty with many staff cuts, both before and during my time there. And the institute, as I said, changed its name and site. Now, unlike other AFRC, BBSRC institutes, we actually only had about 25% co-funding, and that was really difficult. So it was difficult because for us in particular, we didn't have any core money for animals, and we could only use what was available from other people's projects. Later, we were allocated some funding, and then Home Office licenses changed in 1986. Before that, we could take blood samples without requiring a project license. But afterwards, we not only we needed a project license, and that made it more difficult for us to use animals full stop. And it never crossed my mind. And I was in this position of trying to work on immunogenetics, so the MHC essentially, and without necessarily knowing very much about the animals that I was using or having much say over what happened. So it was actually quite tough. And we weren't eligible for research grants at that time. Well, that did change later. And it was not easy for us to have students. So I don't know how I managed really, but I kept going. And by the 1990s, there were much more opportunities. The 80s had initially focused on one region of the genome, and I worked alongside geneticists, but had no idea really what they were doing. They were quantitative geneticists, so they weren't people who were, they were looking at breeding for this and that, mostly animal production traits. And slowly, they began to realize that actually animal health is a really important factor, and that there may be possibilities of harnessing genetics of disease resistance, for example. And so I then started to have major collaborations with the geneticists. So this was an opportunity for me that wouldn't have occurred if I'd been working somewhere else. And it actually then became the absolute major focus of my research for the rest of the time. So we were looking at not just the MHC, but other regions on the genome that might actually affect the immune response. Now, to start with, we were using vaccines and peptides derived from foot and mouth disease virus. We were using a cattle breed cross, which was structured so we knew which animals were which. And as I said, we were looking at hundreds of markers. And we did find significant associations both at the MHC and elsewhere in response to these peptides and vaccines. And then later in the 2000s, there was a technology revolution essentially in genomics. And I know Anne mentioned about collecting data. So actually what I ended up doing is collaborating with bioinformaticians, geneticists, data analysts, all sorts of people that I would never have met necessarily if I'd stayed very much focused on the immunology. And I had a fantastic collaboration with other geneticists at Roslin, with Scottish rural colleges and with Northern Ireland scientists to look at bovine TB, which remains a major issue in the UK and Ireland for reasons that are not entirely clear. And what we did was we set up a case control trial with the help of our Northern Ireland colleagues and looked at about 2,000 animals in herds that were affected by bovine TB, and we divided the animals into infected and non-infected cases. And these were non-infected cases that stayed non-infected over a considerable number of years, and despite exposure within the herd. And we found, in the end, about 3 loci, including the MHC region. And then we replicated that MHC locus, which is not necessarily anything to do with the class 1 or class 2, because there are just so many genes within the MHC region. And so at the end of the day, we had a means of essentially exploiting these findings so that the genetic component could be exploited by breeders, and essentially led to a new tool being developed for sire selection, which is hosted by the Agriculture and Horticulture Development Board. So they are using it, and farmers are making use of the tool as well. So I've probably talked quite a while, so I shall skip over the next. Oh no, I'm not telling you about this. No, I won't. I'm going to skip this because I think I've talked for long enough just to what I did. So what were my other activities going on whilst I was doing all this? I had a daughter. And I worked 80% time, as did my partner actually. And after I came back from maternity leave, I was promoted to the next level, which was my second attempt. So what I would say to you all is, if you don't succeed, just keep going and try again. However, there were issues relating to my working 80% time, as I wanted to now run my own group, and yeah, so I can tell you more about that, but I'm not going to really go over it, except to say I felt supported by the BBSRC and very not supported by the management. So in '94, there was yet another report on women in science, or STEM as it is now. It was called SET at the time, called The Rising Tide, and that encouraged the University of Edinburgh, which we were kind of affiliated to set up a mentoring scheme. Now, at the time, they didn't have enough senior women to actually act as mentors. So my other female colleague, and there were 46, I think there were about 46 senior scientists, and there was me and one other woman in our institute. So that tells you a lot. Anyway, we were both asked if we would volunteer to go onto this mentoring scheme, which we both did. And I learned a lot by doing that, a great amount. It was great to talk to young people. I mentored people, different types of people, a technician, and also a postdoc. And also, the great thing about this scheme was that it had a network for the mentors, which was really, actually, it was like a revelation, suddenly meeting all these other senior women. It was really an absolutely excellent network. Of course, it didn't last. It was for a few years. I was an honorary lecturer in the honours course. I had several students, and I was active in the Career Development Committee. So, and then towards the end, I started to win grants, because we started being able to actually apply for grants. but although they were mainly collaborative ones and through special initiatives and the EU. So then we move forward to the 2000s, and I'll just briefly mention that in 2006 I applied for promotion to the next grade up, and I didn't get it. And actually, I was really supported actually to do that from the management team. Everybody said that I should get it, but I didn't anyway. And then we were working toward, we were really unsustainable. I think this institute would have gone under if we had not come into the university, simply because we didn't have enough core funding, etc., etc. And actually 2007, when David Hume was appointed director, just before we actually moved into the university, the institute's focus changed, and I never ever thought that would happen. You know, I never predicted it. It was like, well, I don't know. It was a great opportunity anyway, and we transferred into the University of Edinburgh, and none of that might have happened. We might not have gone into the vet school. Originally, the idea was that we would go into regeneration medicine because of the Dolly project and the Dolly other such things that were going on at Roslin. And then in 2010, although I was told by my director, oh, I thought you would be happy enough, essentially, being at reader level. And I thought, well, that's fine, but if I'm at reader level, I'm happy with that, but, you know, I'm going to apply anyway. So I was awarded a personal chair in veterinary immunogenetics, and that's me smiling gleefully at the late Prince Philip, who was the Chancellor at the time. And then I became active in the Athena Swan applications, which was led by my other female colleague of long ago, Helen Sang, and in 2017 we won a gold award. And at that time, the Roslin Institute had about 30 women at grade 10. 30% of women at grade 10. So the world had changed. But I have to say that I think some of it, at least, simply reflects the way society has gone over the last 30 years or so. That and I think that is one thing that always occurs to me, is that we don't monitor the effects of doing that kind of application, getting a gold award. What difference does it make? It would be possible to do case control studies, but I don't know of any that have ever been done. And at the time, I also organised a workshop on unconscious bias, because I was convinced that unconscious bias does exist, and I think it absolutely does. But the way in which the universities and institutes, this is perhaps controversial, is that they provide an online course, for example, once, and that's you can tick a box. It's not like that. You should be pushing for it all the time, and you should be going back and checking what policies you have in place. I don't know. I mean, I'm now no longer there, and perhaps Jayne or Guy can tell me a bit more about what's been happening in the last few years. I volunteered to become the editor-in-chief of a journal called Veterinary Immunology and Immunopathology, and that was a wonderful learning exercise. I'd been on the board for lots of years, and then I decided that I would retire, partly because I just didn't want to face writing another grant. And I had many failed grants, apart from all the ones that were successful. So I hope that's going to spark some interest in and questions, etc., in the panel discussion and other things that we could discuss. So I hope that was all right. Thank you.